Okay, this is Franchini. I'll be chairing the next session, which is on prophylactic vaccine. And we have a series of speakers talking about different kinds of vaccine. And I happen to be the first speaker, so I introduce myself. And I work at the National Cancer Institute, and I collaborate with many, many people in this room, as well as others. And what I will tell you today is how we have used the animal model to try to understand how we can, first of all, why a vaccine could work, and what can we do to improve it. So what we have done is to use basically almost exclusively the SIV MAC251 model using the Rosier virus, which is the genetically diverse strain propagated in macaque cells. And over the last 20 years, we have used always the same virus. And whether it was collected at week 8 or 9 or 10, it was from the same culture. And this is highly infectious virus. Is by the oral route, rectal, vaginal, and venous route in macaques. And induces an early and sustained CD4 depletion in gut, and basically most animals really develop AIDS if you wait long enough. So in this model, we have realized after several studies, even early on from the 2005, that if we were using high dose of virus, there is no way that you can actually have a readout of vaccine efficacy. So we moved then later on to using as a vaccine efficacy measurement the pre-exposure risk of SIV MAC251 acquisition in vaccinated and controlled macaques exposed by the rectal or the vaginal route to obliquely doses of SIV MAC251 that transmits approximately very few variants in approximately 30% of the animals. And at the end, we calculate the vaccine efficacy just looking at the controls. Actually, we don't. The statistician does it, looking at the acquisition in controls versus the vaccinated animals. So, as you know, there has been a human trial that has been working, which so far has been ignored, and there are many people that are very skeptical about these results. And this was obtained using the canary pox, which is an heavy pox that can infect the chicken and induce a productive infection, and also kill the animals. But in mammalian cells, this virus induces an abortive infection, so you can actually express an heterologous gene. And this protocol has been developed early on in terms of prime boost approach, which was pioneered by Shulo Ku very early on at the beginning of the vaccine field for HIV. So this protocol, which is around 6 months, in humans, has induced a vaccine efficacy of 31.2%. As I said, many people are skeptical because the number of patients, even if there were 15,000 people, 8,000 people in the vaccinated and 8,000 in the placebo, people are skeptical because when they go back and look at the number, there are very few people that are, if you take off some animals, some individuals from one arm versus the other, but you never do that in human trials. So this is the result, and I think we have to live with that. So, whether you like it or not. And so, the correlate of protection were also interesting and unexpected because it was IgG level to a region that has been largely ignored, which is the V2 region, and CD4 responses, no CD8 responses or very negligible CD8 responses are induced by this vaccine, and antibody-dependent cell toxicity in individuals that had low IgA responses. So what we did in 2005, we used neonate macaques with Martas and showed that this vaccine, giving only 3 ALVAC-SIV, no GP120 boost, so no antibody, really very low level of antibody were induced, elicited a 66% protection in using the comparison of the exposure time in the animals. And at the end of the study, several of these animals remained uninfected. They were challenged later on when they grew up, and there was still some vaccine efficacy. Eventually, vaccine efficacy disappeared, but it was there. What was striking in this study is that there was really no evidence, really no strong evidence of adaptive responses. So this was one of the studies that led really to the performance of the human trial in Thailand, which is called RV144. So what we tried to do in 2016, we actually published that if you now repeat the study that we had done in neonates in Indian rhesus macaque using an interrectal challenge, and we had we were lucky enough to be able to 50% female, 50% male. So this is interrectal challenge. You can see that you can reproduce vaccine efficacy in these animals. Here we had the 27 vaccine and 47 controls, large number of animals necessary if you have a vaccine that is not very effective. So we repeated it in Chinese rhesus macaque because we wanted to see whether it is reproducible. And we used it this time only female, and we did the intravaginal challenge. And you can see that we can reproduce the vaccine efficacy that we have seen in Indian rhesus macaque. So we believe that even if there might be some skepticism, if there was an artifact in the RV144 trial, then we can reproduce the artifact in different studies, okay? Or the possibility is that actually this is really a vaccine effect. So what we did also over the time in the last 10 years is to try to see whether we can improve the vaccine. I will not take you through all the study that we have done for that. We tried to change the adjuvant, we used the MS59. We tried to change the prime, we used the R26. We tried to change the vector, we used the ALVAC. But the best protocol that we obtained at the end always used the same animal model and the same similar challenge, either rectal or vaginal. And what we found is that the best protocol so far is this. It's a shorter protocol, which is only for 3 months, where we give a DNA, 2 immunizations with DNA, and we have done this in collaboration with George Pavlakis and Barbara Felber. And then we use the 1 inoculation only of ALVAC-SIV and 1 GP120 in alum. The dose of alum is very, very important. If you decrease the amount of alum, you don't get the same level of protection. We have a paper going on that. So we use then all these studies to try to understand the correlate of protection. And but before doing that, I just want to show you that we improve vaccine efficacy into 69% with having here a large number of animals that remain protected from infection. What we did then after we took the animals that were protected in here, challenged them again 7 months later, and you can see that you still can monitor vaccine efficacy. And the remaining one, we challenged them again 1.5 years later, and you still can see vaccine efficacy. Eventually, they become infected. Remember, this is a dose that is much higher than what people are exposed to HIV. So, in fact, 30% of the control at each dose. So, but these animals were also vaccinated and exposed. When we did a study in which we vaccinated the animal, waited 1 year, and challenged them, we did not see vaccine protection. So it tells you that there is probably a range of months that the vaccine protection lasts. So we used, as a technical approach, a variety of systems, a variety of studies to really try to see, to understand the correlate of protection. So one obviously was traditional immunological assay. The other one was system serology, transcriptome analysis, microbiome, and so forth. And I will only focus on this and in part on the transcriptome because of time. But at the end, all the data that I show you, or the conclusion that I show you, are really derived from more than 300 animals immunized with ALVAC-SIV-based vaccine, and all challenged with the same virus. So what we found is several correlates. So we found correlate of decreased risk, and what is highlighted here are those that are more frequently found in the different studies. And but also we find correlate of increased risk, which we don't really consider. And by the way, we should really study more in non-human primate. I think that when we challenge our animals, we challenge them in a way that all the controls become infected. I think we should start to do studies in which we challenge them with doses that will not infect all the all the controls, so we can start to see enhancement. Because if you do that, you're going to see enhancement. We are not in condition to measure enhancement the way in which we do the experiment. So as I said, we have all this, myeloid cells is really very strong signature, and I will tell you what I think is happening in here. But also you find NK cells. For example, if you challenge indirectly, a correlate is NKp44 IL-17 positive cells. But if you now challenge vaginally, these cells are not very frequent in the vagina. Now you find the NKG2A CD107 positive that correlate. And then there are also Th1 cells which correlate very nicely. V2 antibody, I don't have time to go through all of them. I just will focus on this aspect, which is novel, because by doing these studies, we found this novel and unexpected immune correlate in myeloid cells. So the data that we had suggested that we were dealing with innate monocyte memory, which is also defined as the trained immunity, which is has been described approximately 7-8 years ago, and and is obviously different from the classical immune memory that is based on a rearrangement of the BCR in B cells or the TCR in T cells. Is specific and is long term, but this is actually doesn't require gene mutation, is non-specific, and is a sustained change which is lasting in cells, which is and usually is in epigenetic changes that induces this lasting memory. So the trained immunity is an ancient innate immune mechanism that has been found in plants, vertebrates, cockroaches, shrimps, and which is interesting. But the cockroaches are extremely successful, as you know, and the insects. I mean, they are everywhere. They certainly are more than us, and this immunity has been shown in experiments in mice that were, for example, stimulated, let's say, with the Candida, and then they were challenged with Staphylococcus aureus and did much better. And that's true for all of these immunogen. And also from human studies, epidemiological studies that show that if you children, neonates that were immunized very early on with the BCG, died at a decreased mortality for any cause over time. Same story for people that old like me that were vaccinated with the smallpox vaccine. Apparently, they have a decreased mortality as compared to people that were not vaccinated. So how does this immunity work? So it looks like that what is happening in monocytes, you have a metabolic switch from oxidative phosphorylation to glycolysis, which results in the accumulation of substrate which changes the epigenome. Just said very simply, there are a lot of papers in this, and you probably can, you will have to look at them. And as I told you, this correlate with monocytes was the most consistent one in our study. For example, we see a nice correlation. This is the study in which we had 70% almost protection in female macaque. You have it in another study in female macaque, and another independent study in male macaque. So it keeps coming up. So I believe that is probably true. And in addition to this, when we did the collaboration with Brandon, and we look at the transmitted variant, you can see that the CXCR4 monocytes, which are newly aggressed monocytes from the bone marrow, they still have an upregulation of the CXCR4, correlate also with the number of transmitted variants. So if you have a higher number of these cells, you have fewer transmitted variants or don't become infected at all. So a feature of our vaccines that we used in here is that the ALVAC preferentially infect monocytes. So when we look, and we did a collaboration with Cindy Dunbar, that is here in the audience, in which we looked at the ability of the different modality, the different components of our vaccine to engage monocytes. And you can see in here that you take alum alone, this is the absolute number of the monocytes in the blood of one animal, which is a barcoded animal. And you can see in here that the alum is actually increasing the number of the monocytes. The GP120 plus alum is also increasing the monocytes, and so is the ALVAC-SIV by itself, as it is known that the ALVAC is able to activate the inflammasome. And so therefore, the vaccine modality that we have used and combined together in here induces the inflammasome and IL-1 beta, probably because it's engaging the monocytes, all of them. In addition to that, we also proved that with Rafik Sekali and Slim Furati in animals that were immunized with the RV144-like vaccine, that you increase the number, but you obviously, as you would expect, you also increase the transcriptome in these monocytes. And these red genes are really genes that are expressed at higher level in animals that are protected. So what we think is going on is that this modality, the combination of this modality, is inducing IL-1 beta, and we have evidence of that, which is able to induce emergency myelopoiesis, and then egress of these cells, which are probably epigenetically changed in the bone marrow, even before they come in the periphery, which then can give you a better response to a pathogen. And so you have to assume that because monocytes have, and they become long-lasting. Monocytes are continuously produced by the bone marrow, and their half-life is basically a day in the blood. So you have to assume that if you have this correlation of monocytes that you have induced by vaccination, you have memory, then they have to change, and they have to be able to live longer. So you can have two different kinds of monocytes. One is when you're a metabolic switch from oxidative phosphorylation to glycolysis, you create a pro-inflammatory monocyte, which is defined usually as strained immunity. But you can also induce, and this has been seen in sepsis, you can also induce monocytes which can make immune suppressive cytokines, which can then block the immune response. And these are anti-inflammatory monocytes that are really important, again, as I said, in sepsis. So what is the protective response against SIV in our case? I personally believe that it is anti-inflammatory monocytes, and I'll show you why I believe that. And the existence of this anti-inflammatory monocyte mechanistically has been shown very nicely in this paper, a recent paper from 2019, which they show that bone marrow cells are making IGF-2 that are able now to induce the PD-L1 on the monocytes, and then which in turn engage Treg cells. So here you have a different mediator. IL-1 beta is inducing trained immunity, while instead IGF-2 seems to be inducing anti-inflammatory monocytes. So what we want to do is in the future is to try to define them. These studies are ongoing, and we have started just to do some of the studies. I just show you one example. We have others, but we didn't analyze all the data yet. This is looking at the lymphocytes or the monocytes before and then after immunization. You can see here, if you look at the enhancer of the IL-10, you have an opening of the chromatin. So again, we have to look at the whole genome of these animals by doing the ALVAC-SIV, which we are doing, and it will take quite some time and a lot of informatic analysis to really try to find out what are the genes that might be important in addition possibly to IL-10. Okay, so the monocyte function, it is well known, is decreasing with aging. So you can see that typically in older people above 65, you have an increase in CD16 monocytes, which are the more pro-inflammatory monocytes. They have low expression of MHC class 1. They don't respond very well to TLR stimulation. They make less, they make less IL-1 beta, and so forth. So what we try to do is to see whether we could see the same effect in macaques. And here we took a cohort of animals, young and old. So young is around 3 years old. Older is the average age is 7.5. And I have to say that we are repeating now this experiment with animals that are 15 years old to increase the difference between the young and the old. And you can see in here that if you look at the ability of the cells in vitro to respond to stimulation, here you have the old animals, and here you have the young animals. You can see that there is quite a difference in the ability of the young, the cells from the young animals to the old animals to really respond to different stimulation. So what is seen in humans is also seen in macaques. So then we immunize older animals to really see whether we could get protection. And you can see in here that you have a strong trend, but you don't really have significant difference in the acquisition. And is 0.1. I mean, you do see some vaccine effect probably, but it's not. And we reproduce it also by using IGF-1, and it's the same. So vaccine efficacy decreases. So one of the questions is why? And so we started to look, we looked at, first of all, whether there was a different susceptibility of young macaque versus old macaque to SIV acquisition. And you can see in here that there isn't. They acquire SIV, these are naive control. They acquire the virus in the same way, so this difference in the vaccine is not due to a different susceptibility of the animals, but rather to the fact that probably this vaccine was not as effective. And so the hypothesis in here is that this vaccine was not effective because monocytes don't work well in older people. or animals in this case. So we did look at the number of monocytes. These are the classical monocytes that correlate with acquisition in young animals. You can see that they are not less, they are more, but they don't produce the equivalent amount of CCL2. So this is the difference between the post and the pre and the post. You can see that in the young macaques, the level of CCL2, which is mainly produced by these cells, is increased in young macaque, but is actually decreased in older macaques. And also, these cells don't correlate with acquisition in the older macaque, as you would expect. And there are other qualitative differences between the young and the old. One is, for example, is the production of IL-8. It's produced as a pro-inflammatory cytokines that calls in a lot of other inflammatory cells. And you can see in here that it's increased in older macaque, and it does correlate with an increased acquisition. So the animals that have high level of IL-8 are the animals that acquire the virus faster. And in addition to that, these animals are the older animals also make less IL-10, significantly less IL-10. So the landscape that you have, the immunological landscape in young and old is quite different. So it's something that we need to consider when we do vaccine study. Also, is the age of macaque. It's not just taking animals from here and there and not matching them for age is probably not wise. So if you look also at the transcriptome of these animals, here is the baseline transcriptome. You can see that the triangle are all the pre for the old, which are the red, and the young that are blue. And you can see that when you vaccinate them, they respond very different to the vaccine. You see that in the old, you have clearly a different way in which the transcriptome is actually activated. So basically, we definitely have seen an age-biased vaccine efficacy. We need to, and this actually confirms that probably the correlate of protection that we have, which is monocyte, is probably correct, and which it's useful. How we can actually revert it is something that we are trying to investigate. And finally, I just want to leave you with this. We found, as I said, a variety of correlate of acquisition, and all of them are correlating also with the monocytes. So it's true, it's all correlate with correlate. We don't have direct evidence. We need to do more direct experiments to really prove that. We need, for example, to immunize animals, deplete the monocytes and the macrophages with the clotronate to really see whether we lose protection. We need to deplete NK cells. So I think that the field needs a lot of these direct experiments rather than just correlation experiment, and that's the direction of where we're going. But another question that comes to mind immediately, okay, so who is actually educating these monocytes? And there was a recent paper from Belcade, Askenase and Belcade, that really shows that NK cells, resident bone marrow NK cells, by producing interferon gamma, that usually we think about interferon gamma as an effector molecule, is actually able to educate monocytes to become anti-inflammatory. And in this vaccine, we also have a very early gamma interferon signature that we see. So I stop in here. And of course, there is the issue that we are immunizing systemically and we get mucosal responses. So how is this occurring? So it could be chemokine and cytokines, but we are looking now also at mirror RNA to see whether there could be messenger from the CD14 monocytes or other cells that might be important. Okay, so this is, it took a village literally to do all this work. And these are the people in my lab that have been working very hard and they've been all very successful. And these are all our collaborators, and I'm sure that I forgot a few of them. Thank you. Questions? Yes.
Thank you. I think what the study nicely shows is, or at least brings up for discussion, is that our constant quest in finding the immune correlate of protection may also not be true, because Lewis has shown that there's a very unique T-cell response that is clearly associated with protection in the CMV vaccine. You have found a potential role for monocytes in protective immunity, and then there are all these approaches that are geared at eliciting broadly neutralizing antibodies, or the RV144 trial with Fc-mediated effector function of antibodies. So I think that's something else we should consider in future vaccine studies, that we should not always look just as what is already established. We should be open in exploring different mediators of protection and different vaccine strategies, and be open to testing those also in clinical trials.
Yeah, and I think also that probably different vaccine will elicit different correlate of protection, because they probably will. I mean, the CMV clearly is a replicating virus. You don't protect from infection in the CMV, but you can control viral replication. Here, you can't control viral replication, but you protect from infection. So it's clearly we are working a different compartment in the immune system.